Welcome to the presentation of interleukin-10 and tumor necrosis factor alpha serum levels in chronic periodontitis with and without diabetes, presented by Al-Azhar, Assistant Lecturer of Biochemistry at the Faculty of Pharmacy, Badr University in Cairo, under supervision of Dr. Abir Ibrahim, Professor of Biochemistry at the Faculty of Pharmacy, Girls Al-Azhar University, Dr. Khalda Amr, Professor and Head of Medical Molecular Genetics Department at the National Research Center, and Dr. Maha Rashid, researcher at the Orodental Genetics Department, Division of Human Genetics and Genome Research, National Research Center. The outline of this presentation includes firstly the background, in which we will speak about the chronic periodontitis, followed by chronic periodontitis systemic associations and the chronic periodontitis and diabetes. Then we will speak about interleukin-10 and tumor necrosis factor alpha, followed by the objective of the study, then the subjects and methods and results and conclusion. The chronic periodontitis. It is the more severe and destructive form of periodontal diseases. It compromises the tooth supporting apparatus, which is the gingiva, the cementum, the periodontal ligament, and the alveolar bone. It is the sixth most common disease worldwide. It adds a substantial economic burden and has a negative impact on the quality of life. The chronic periodontitis is characterized by infiltration of inflammatory cells, destruction of connective tissue attachment, and progressive alveolar bone resorption. Clinically, the patients present with the deepening of normal gingival sulcus, which is the soft tissue pockets between the teeth and the gingiva. In severe cases, it can result in loosening of the teeth, occasional pain and discomfort, impaired mastication, and eventual tooth loss. This diagram represents the development of periodontitis from a healthy periodontium, which is initially caused due to the microbial dysbiosis occurring in the periodontal biofilm in a susceptible host with the periodontal disease risk factors. When the balance between the resistance and the risk factors is altered, periodontitis develops. The chronic periodontitis systemic association. The periodontal infections have long been indicated as a potential risk factor for several systemic diseases. This occurs due to the proximity of the periodontal biofilm to the gingival blood microcirculation. Thus, the periodontal pockets act as a reservoir of microbial pathogens and their products, as well as inflammatory mediators and immunocomplexes, which may disseminate to other sides of the body through the gingival circulation. The periodontitis starts at the junction of the tooth and the gingiva, where there is increased tissue breakdown products and increased inflammatory mediators, which disseminates into the circulation, causing a systemic inflammation. The systemic inflammation increases the effects of any ongoing inflammation in any distant organ. As we can see, it promotes atherosclerosis and increased inflammation-induced pregnancy complication and other diseases. The mechanism for the periodontitis systemic association starts with the bacterial challenge in the periodontal tissue, causing a host response which is usually exaggerated and hyperinflammatory in a person with the periodontal disease risk factors. And the prolonged host response causes the periodontal disease, which is in this case periodontitis, leading to a chronic low-grade inflammation, which exacerbates any ongoing inflammation in distant organ, leading to increased risks for atherosclerosis, diabetic complication, adverse pregnancy outcomes, respiratory diseases, and neurodegenerative diseases. Chronic periodontitis and diabetes. Inflammation is a central feature of the pathogenesis of both diabetes and periodontitis, where diabetes is a major risk factor for periodontitis, and the susceptibility to periodontitis is increased threefold in people with diabetes. It's also important to note that diabetes exacerbates periodontitis and vice versa. The mechanisms that underpins the links between these two conditions are not completely understood, but they involve aspects of immune functioning, neutrophil activity, and cytokine biology. There is emerging evidence to support the existence of a two-way relationship with diabetes increasing the risk for periodontitis and the periodontal inflammation negatively affecting the glycemic control. Interleukin-10. Interleukin-10 is a cytokine characterized by its powerful anti-inflammatory properties. It acts to preserve the host from the aggressive host response against foreign bodies, thereby maintaining homeostasis. Tumor necrosis factor alpha, or TNF-alpha, is an inflammatory cytokine involved in the systemic inflammation. Its primary role is the regulation of immune cells. It is responsible for stimulating the acute phase reaction. It also induces the apoptotic cell death. It inhibits tumor genesis and viral replication and is thought to be implicated in the periodontitis pathogenesis. Thus, we could conclude that the balance between TNF-alpha and interleukin-10 is crucial for homeostasis. The objective. Firstly, to evaluate the serum levels of interleukin-10 and TNF-alpha in chronic periodontitis and diabetic chronic periodontitis patients and to compare them with the healthy individuals. To correlate the serum levels of interleukin-10 and TNF-alpha with some clinical parameters of periodontal disease. To demonstrate the inflammatory status of Egyptian patients with chronic periodontitis and chronic periodontitis diabetic patients. The subjects and methods. The study subjects were selected from the outpatient clinics of the National Research Center between the period of July and December 2018. The study included a total number of 50 individuals divided into 3 groups. The first was the control, made of 10 individuals. Then the 2 groups of chronic periodontitis and diabetic chronic periodontitis. Each was made of 20 patients. All subjects completed an in-person interview on current oral health status, history of dental diseases, procedures, diabetes history, and occupation. The inclusion criteria included, firstly, both genders of systemically healthy individuals according to Burkitt's Oral Medicine Health History Questionnaire, patients who agreed to sign a written informed consent according to the World Medical Association Declaration of Helsinki, and an untreated periodontal disease in the previous 3 months. The exclusion criteria of the selected patients was any systemic inflammatory or immune diseases which can alter the inflammatory status of the patients, pregnant and lactating females, also females taking oral contraceptive drugs, and subjects who had received antibiotics or treatment for periodontal disease within the previous 3 months. All subjects completed the following: full history taking, clinical measurements recording, including probing pocket depth, gingival recession and clinical attachment loss, blood sample collection and then serum separation, biochemical investigation of interleukin-10 and tumor necrosis factor alpha levels in the serum using commercial kits of sandwich ELISA. The methods. For blood collection and serum separation, the serum was separated from a 3 ml blood sample, then allocated in labeled Eppendorfs and then stored frozen at -40 degrees Celsius for further analysis. As for the biochemical analysis, prior to the analysis, the samples were brought to room temperature, mixed 3 times using vortex, and then the supernatants were used for the analysis. For interleukin-10, a commercial solid-phase sandwich enzyme-linked immunosorbent assay was used, and for the TNF-alpha in the serum, another commercial solid-phase sandwich enzyme-linked immunosorbent assay was used too. The results. The clinical parameters for the diabetic chronic periodontitis and the chronic periodontitis, the clinical parameters was significantly increased as compared to controls only and not when compared to each other. For interleukin-10, the controls showed the highest mean plus or minus standard deviation, and in the case of chronic periodontitis and diabetic chronic periodontitis, the mean plus or minus standard deviation was significantly decreased. While for the tumor necrosis factor alpha, the lowest mean plus or minus standard deviation was demonstrated by the control group. As for the chronic periodontitis and diabetic chronic periodontitis, both were significantly increased as compared to controls only. This is a different representation for the mean plus or minus standard deviation of the concentration of both interleukin-10 and tumor necrosis factor alpha, where the control had the least amount of tumor necrosis factor and the highest amount of interleukin-10. Interleukin-10 levels decreased in both chronic periodontitis and diabetic chronic periodontitis, while the TNF-alpha levels increased in both chronic and diabetic chronic periodontitis. The correlations of TNF-alpha serum levels and the clinical attachment loss in the studied groups. We can see that there were positive correlations between TNF-alpha and clinical attachment loss in both groups, although the correlation was not very strong, but it was significant. The correlations between interleukin-10 and TNF-alpha levels in the studied groups, we can see that there were strong negative correlations between the TNF-alpha and interleukin-10 in all of the studied groups, firstly the control, then the chronic periodontitis and diabetic chronic periodontitis as shown in the figures. Finally, the conclusion. Chronic periodontitis with and without diabetes contributes to an increased serum inflammatory burden, which predisposes other inflammation-related diseases. The imbalance between interleukin-10 and TNF-alpha is involved in the chronic periodontitis pathogenesis. The interleukin-10 and TNF-alpha have a great potential clinical value for monitoring the inflammatory status of patients and in identifying population with susceptibility to inflammatory diseases. Thank you.